Thank you for the opportunity, and I'm going to tell you a little bit about the exRNA pipeline, which is the RNA-seq processing pipeline of the ERCC. You already heard briefly about this from Rob Kitchen and Matt Roth just a little earlier, so I'll give you a little bit more details. As Roger says, I am at Yale, and I am part of the data integration and analysis component of the DMRR that Matt told you about. And we've developed this RNA-seq processing pipeline for extracellular RNA data that was generated by the ERCC. This pipeline excerpt that you heard about earlier was published in 2019, and this was done together with Rob Kitchen when he was here at Yale. Currently, at the time when the ERCC was developed, ERCC has generated lots of data set, but there wasn't any tools to analyze RNA-seq specifically in the context of extracellular RNA. So me and Rob developed this pipeline with the eye towards focusing on data, RNA-seq data generated by the consortium. And one of the big issues is in order to be able to integrate and compare data sets across the consortium, we wanted to be able to uniformly process all the data. And that was part of the motivation for generating this pipeline. At this stage, we've analyzed the pipeline can also be used on other RNA-seq, small RNA-seq data sets, specifically in the context of cellular RNA-seq data too. But and all the more than 6,000, approximately 6,000 extracellular RNA-seq data sets that are in the exRNA atlas have all been uniformly processed using this pipeline. So if one wants to use the pipeline, you can either go access it through genbury.org, what Matt was telling you about, where the tools are available. There's a GUI interface for running exRNA, where you can upload your files and run it on the cluster at Baylor using Genbury. For people that are more inclined to run it in a more custom process, we have a GitHub repository where you can download the source code or run it or install it locally via Docker image, and then you can run it locally and processing the data on your own local cluster or own local computer. So you've seen a version of the slide before from both Rob and Matt. So as Rob mentioned earlier, we developed this pipeline specifically for processing extracellular RNA. And we, because at that time there wasn't other comparable standardized pipelines, we adopted a very conservative approach for analyzing extracellular RNA, because we were very worried about potential contamination and potential other sources for RNA, such as DNA and so on. So we built this pipeline where we have this elaborate pre-processing filtering step, removing reads of low quality, removing reads that potentially corresponding to known laboratory contaminants, removing endogenous ribosomal sequences that we tend to be less interested in. And then we map the reads against the annotations of the endogenous genome. By default, the pipeline can work on both human and mouse annotations. And also, as Roger mentioned a little earlier, the quality of the output of the pipeline is contingent on the quality of the annotations. So for genomes such as the human and mouse genome, where the high quality annotations exist, this is very successful. For novel genomes or genomes that aren't annotated quite as accurately, one could use this pipeline, except then the limitation is based on the annotations. So by default, we quantify the known annotations using the order that I show on the slide. So microRNAs, which tend to trust the annotations best, then tRNAs, pRNAs, and so forth. But for a data set where you are specifically targeting a small RNA of a different type, one could adjust the order which the annotations are quantified. All the data in the exRNA atlas is processed uniformly with this default annotation order. After we've we've quantified all the endogenous RNAs that are present within the organism, which in the atlas is mostly human and a small amount of mouse data too. We do a second pass alignment against the host genome to remove reads that potentially we don't want to use for quantification, but potentially come from the host genome. And then we do a sort of steps 2 and 3 in the pipeline where we map against exogenous genomes. First known exogenous microRNAs, exogenous ribosomal RNAs, and then the full genome sequences for a whole host of exogenous genome sequences. And all the data in the exRNA atlas by default runs to the end of the pipeline, but when you run it sort of either Genbury or sort of your own download, you could truncate the pipeline earlier in the step if one's only interested in endogenous quantifications, and if you're not particularly interested in the exogenous, because the later steps in the pipeline tend to be the most time consuming. So this is Rob sort of showed a version of this, so I won't go into this in too much detail. So this is the pipeline on the left-hand side, and sort of just zooming in on the right-hand side, this shows the effects of the various steps along the pipeline if you basically leave out a step. So I'll just sort of highlight one step is if we remove the sort of alignment against known laboratory contaminants, this UNIVAC contamination. So if we leave this out the step in the pipeline, what you find is that the number of reads, so the first column shows where the reads, so if we start with 100% of reads, where the reads land up mapping at the various stages along the pipeline. So if you see, this is, I think, a typical plasma sample. We see most of the reads map to endogenous, the majority are microRNAs and so on. And we get a small fraction of reads, around 4.4%, that land up mapping to exogenous genomes. But if we leave out the step in the pipeline of mapping to known contaminants, then we find a significant increase in the amount of reads that map to these exogenous genomes. So this shows the effect that if one didn't filter out known contaminants, you would potentially misidentify more exogenous genome sequence reads that are present in the, as part of the output than one should correctly identify. There are other tools for analyzing small RNA-seq data, such as miRDeep. And in the paper, we sort of quantify, correlate the quantifications of microRNAs between our excerpt pipeline and miRDeep, and we get very high concordance. This just sort of shows what the sort of relative distribution of biotypes one obtains for different representative biofluids, human biofluids. And you can see that there is variation between specific samples, but you see that the distribution of biotypes varies significantly between different biofluids. So for example, in the urine, we get a significant fraction of these green, which is the tRNAs. In saliva, we get a lot of red, which are these exogenous, mostly bacterial sequences. In serum and plasma, we get a large fraction of microRNA sequences. And even though you get a variation between specific samples from specific individuals, different biofluids tend to be have a sort of specific signature that is distinct from most other biofluids. So as part of the excerpt pipeline, when one runs it, it also analyzes the reads in terms of, as I mentioned, in terms of the reads that map to exogenous genomes. So we map against databases of known exogenous RNAs, such as microRNAs in miRBase and ribosomal sequences in RDB. And then we map against not all, but a reasonably large complement of full genome sequences. So at the time of the publication, all bacterial genomes, all fungal genomes, plants, protists, and then a subset of vertebrate that are likely things that would be in the diets of humans. So things like cow and other representative. And then we also map against all the viral genomes in NCBI as of the time of the publication. So when we map against these exogenous genomes, this is just a diagram that sort of shows how the algorithm works. So we map independently the reads against all the genomes. Now, as you might expect, many genomes share a high degree of sequence similarity between them. So read number 1, the first row in this thing, if we look at the alignments, we find that it aligns against genome A, genome B, genome C, and genome D. So what we do is we take the phylogenetic tree of organisms and we assign the read to the node in the tree that's most consistent with the genomes A, B, C, and D, which is this node over here in the tree. Similarly, for read number 2, which aligns against genomes A and B, it gets assigned not to the node at the end, but the node that is consistent with A and B. And this is the strategy we use to where we assign reads that align to multiple genomes. If a read maps just to a single genome, then we just assign it to that specific organism. So here's an example of the output of a saliva sample from David Wong's lab at UCLA. So David Wong sort of generated a whole bunch of saliva samples. And when we do this sort of assignment of reads mapping to bacterial genomes, and we sort of look at the reads assignment to the phylogenetic tree corresponding to bacteria, what we see is we get the Streptococcus as sort of the most biggest contribution. Now, what's interesting is when we do the alignments, we can construct this phylogenetic tree using reads that map to exogenous ribosomal sequences. And we could also construct the phylogenetic tree using reads that map to exogenous exogenous genome sequences. So these are completely disjoint set of reads. And the fact that we actually basically get Streptococcus, the phylogenetic tree recapitulated using these two disjoint set of data from the same individual gives greater credence the fact that we're actually really observing sort of this Streptococcus is present in this specific saliva sample. I'll show you just as a sort of comparison, this is a second saliva sample from David Wong's lab. And in this context, again, using the reads that map to genome reads and the reads that map to exogenous ribosomal sequences, we get the same phylogenetic tree, and we find this Prevotella is present. And this Prevotella is another bacterial species that's part of the oral microbiome. So both this and Streptococcus make sense that they would be present in a saliva sample. And these tree outputs and things are all part of the sort of default output of the excerpt pipeline. Lastly, as part of the pipeline, when one runs it, we calculate a number of quality control metrics. And this is applied to all the data that's being processed through the excerpt pipeline that's in the exRNA atlas generated as part of ERCC1. All the data is there. We flag the data that meets the criteria. And currently, more than 95% of the data in the atlas sort of meets this criteria. I'm not going to go into too much detail because I think I'm probably running out of time. But these are sort of very relaxed, but sort of criteria sort of to identify sort of samples that are potentially of poorer quality. So just in a summary, I've give you a brief introduction to this excerpt pipeline for our RNA-seq processing pipeline for extracellular RNA-seq data. You can go access the pipeline either via our GitHub or run it through the graphical user interface at genbury.org. And as part of the pipeline, we calculate these QC metrics for all data that's processed through the pipeline. So just as part of the acknowledgement, thank the group that was at Yale. Rob used to be at Yale, and there's a bunch of people that have worked on the pipeline. The group at Baylor with Matt and company, and then many other members of the ERCC. So thank you for your attention.